coronavirus, assembly, and budding, and all the other things. So, we can divide up the viral life cycle into, or viral replication cycle, into all these different phases. If you're just thinking about how do we get a virion, there are actually all these steps involved, and there are special proteins that are involved in each one of these things, sometimes only one, sometimes multiple. So let's break it down. Yeah. First, you've got your expression. Each of the proteins has to be made into a protein from a subgenomic RNA. That's just how it works for coronaviruses. That's going to involve your translation, but also things like glycosylation. We've got several glycoproteins here. The spike, the M, and there are a couple other minor proteins that are not essential that also get glycosylated. HE is a protein you don't find in SARS coronavirus, you find it in others, but it would be glycosylated if it was there. So, yeah, if that makes you happy, now you know. Also, acylation, that's attaching fatty acids to molecules, and that's going to happen to S and E. And phosphorylation, that's going to happen to N. And you'll also have these things going through chaperones and stuff like that. HSP70 is one of the big chaperones that's involved. And those are just going to help refold, unfold, fix misfolds, that sort of thing. And these other things like the glycosylation, the acylation, those also help with folding. So that's all in expression. And so when we're talking about expression, we're talking about a lot of things that mostly the host cell is doing. And all the virus is doing is providing a template which has the right signals for the host to recognize and do the stuff to. Yeah. All right. Then you've got transport, which is a big thing. Because a cell is enormous compared to a virus and super enormous compared to a virus protein. It's like the difference in size between you and the Texas A&M campus, like East and West campus both. And so if you imagine, how easy would it be to find one other person of your size if they could be anywhere else on the campus, like in any room, yeah, or on any sidewalk anywhere? One, it's too hot for that kind of nonsense here in Texas. I'm pretty sure of that. And two, yeah, you don't have that kind of time. You're a busy person. So it's the same thing for the virus. It's got to rely on cellular trafficking pathways to concentrate all of its forces at the point of attack, and that's where it's going to form a virion and bud and make other people sick. Good job, virus. So yeah, transport. That's also going to involve a lot of cellular machinery. That's a very cell-focused process. And the virus is going to do nothing to modify that process. It's just going to basically forge credentials and hop on the system, hop on the right train with a fake ticket. Yeah. Terrible viruses. Next, you've got two things that are often confused and lumped but are actually separate. So you've got packaging, which means putting the genome and only the genome into the virion. Because a virus, like a coronavirus, is going to make lots of copies of its subgenomic RNA and very few copies of its full-length genome. But the only thing that can start a full infection is the piece of RNA that has all the genes on it, and we call that the genome. So you got to get the genome into the virion, otherwise you're launching out virions with subgenomic RNAs and they can't start an infection. You are basically wasting a perfectly good subgenomic RNA for no gain whatsoever. So don't do that. Yeah, and the virus tries not to. So packaging is the specific interaction with the genome and the structural components. And the N and M proteins are going to be responsible for that. Assembly is bringing together all the components. So not just N, M, and the genome. E is going to be there at the spot, and S is also going to be there at the spot. These are the structural proteins. These are the ones that all have to come together. And so that assembly is going to be partly driven by transport and partly driven by interactions among the proteins themselves. And so here's a list of some of the proteins that will come together. For example, two M's will actually dimerize. M you'll only find as a dimer in cells. And it dimerizes through the transmembrane and also through the tail section of M. They will both help with that. The C-terminal domain of the nucleoprotein is also a dimer forming thing. So you get a lot of pairs of things, and E will all form a little pore of 5 of these E's. And we don't know if that's important or if that's just a thing that it does when you get it all by itself, but it's a thing it can do. It's part of its repertoire, so that's fine. We respect that. Budding is after all the pieces come together. They actually come together like a piece of IKEA furniture. If you've ever been to a big city that has an IKEA, flat packed, just like that. And then they come together and you can see them flat packed, like a big thick bit of membrane that's still totally flat, and it'll sometimes even have spikes sticking out of it. And then what happens is it's going to bend up like that and bend around and then sit back down, and then the last step is going to be scission. So budding is bulging the membrane, but it's not breaking free. It's just creating a bulge in the membrane that you can then go on to cut free at this process called scission. Budding is going to be driven by M, the membrane protein. M is going to have help from its friends. It's going to get a little assist from S and N, possibly E, and those are going to help it to do its job, but M's got to carry the load for the most part, because M goes through the membrane 3 times. So think of it as having 3 ways to push against the membrane. Actually, it's just one big thick way to push against the membrane, but yeah, there's a lot of M as well. For every one of these spikes, that's a trimer, there's going to be 3 spikes there. There's a little grid, and you're going to have 16 pairs of M's. So you'll have 32 M's surrounding each spike. And that's the basic structural unit. And then hooked up underneath, you'll have a bunch of N dimers as well. All right, so those are the steps of coronavirus structural protein, the life of the structural proteins. This is what the question's about, people. Yeah. So I'm going to pause there, and then we're going to go through each of the proteins separately, and then I'm going to have separate little videos where we actually look at an alignment of some of these proteins. We look for which parts are conserved. We're going to learn the way that SARS-CoV-2 does it, but then we're going to learn, does every coronavirus do it this way? You know, how important is this in general, or is this something that's only important for SARS? So, hope you'll stay tuned. It's going to be great. All right.